Thanks, Dr. Bold. It's been many years, so.
I didn't say what year we graduated.
Yeah, thank you for that, but I'll date myself through this anyways. Okay, I have no disclosures. So a little bit about the title. So what I'm really interested in is this relationship between chronic inflammation and cancer, and you'll see that theme come through as I go through my talk. We'll discuss a few of the tools and concepts that we're kind of using to try to figure out this relationship. And I'm going to start with a little bit about me. None of you know me very well. Rick knows me the best. I've seen some former graduates that I've worked with through the years, and then my dear friend, Dr. FASCRS. Thank you for inviting me. Thank you and Dr. Farmer for inviting me here. I've had a wonderful visit. So I actually have a very kind of atypical pathway to where I am now. I actually went to a conservatory of music for my undergrad and started in piano performance, so pretty atypical. When I was there, the people in the conservatory all had to take a basic science class. So thinking that I was actually going to end up in Carnegie Hall, I took genetics as a freshman, thinking it wouldn't matter if I got a D or even if I failed, because it didn't really matter. And I did really well in the class, and thank God I did, because 2 years later I realized I wasn't good enough to go to Carnegie Hall, and I ended up majoring in psychobiology, which was basically what we now call neuroscience, and the rest you'll hear about in a minute. So Dr. Bold is correct. We both ended up at Stanford. I had a weak research background and a probably weaker medical background, coming from mostly learning about piano pedagogy and music theory, but somehow I got through. I did HIV research there, and actually it was there that I really got interested in this relationship between inflammation and cancer. You remember probably Dr. Oberhelman, so I rotated on his service, and there was a lady who was 36 years old that was going to the operating room for resection of a colon cancer. She didn't have a bunch of polyps or anything, nothing familial that could be sorted out. And Dr. Oberhelman, when I said, hey, she's only 36, she's a little bit more than a decade older than me. Why is she going to the operating room? And he said, well, all we know is that she's had ulcerative colitis for about 16 years. We don't know what that has to do with this, and our job as surgeons is to take it out. So eventually, I ended up in this field where my kids used to ask me, what do you do, Mom? And I used to tell them, this is part of what we do. We look in people's bottoms, and so they used to proudly proclaim, my mom's a butt doctor. But they also said, I never want to do what you do. Learning objectives for today. We're going to talk about inflammation-associated malignancies. We're going to talk about some novel tools and models that we've developed to try to understand these relationships. And we'll look at passive members, formerly passive members of the microenvironment, and see how powerful they are in different contexts and how they can contribute to the pathogenesis of the disease, or potentially how we can use them potentially as tools to mitigate disease and disease progression. And we'll do that looking at one very powerful tool, reprogramming technology. And I'll show you a very elegant model that we're just working on developing to ferret out some of these relationships. So most of you know that colon cancer is the third most common cause of cancer in this country, and the third most common cause of cancer death. And most of us have been taught by my friend Eric Ferron that there's a number of mutations that happen during the pathogenesis of sporadic colon cancer. First, you get a polyp, and you get some associated mutations, often in the APC gene, the Wnt signaling pathway, RAS. This has largely been thought now to be replaced by SMAD or TGF-beta signaling. And then finally, you get P53 and invasive cancer and metastasis. And that's okay for sporadic colon cancer, which is some of what we study and most of what we operate on. But it's not so well understood for colitis and colitis-associated cancers. Most of you know that these diseases affect about 1 million patients in the United States, so Crohn's colitis, ulcerative colitis, and these patients often are affected in young adulthood, just like the patient that I talked to you about that I met as a medical student, and they have multiple manifestations, anywhere from mouth to anus to skin. And I would say that even in our hands, we would say that even their brain is affected with their pain threshold and so forth. But they often present with different kinds of bowel manifestations, fibrosing disease, stenosing, and bloody diarrhea. The episodes are chronic, remitting. We don't understand what sets them off. We don't understand what causes the primary disease. In the world, about 18% of the world's cancers are associated with some kind of infectious inflammatory event. And now we're thinking that even more are associated, whether it's smoking and tobacco, dietary supplements or things that we eat. And even obesity now is considered a low-grade inflammatory event, which can be associated with different types of cancers. They may be led by tumor promoters, or there might be infiltration of weird immune cells, or maybe your immune system isn't able to determine that there is an event that happened that's oncogenic and manage it. But these events are poorly understood in the case of colitis and then colitis-associated cancer. What we do know is that, and this is very vague, that there's for the initiation of inflammatory bowel diseases, there's some kind of relationship between your genetic susceptibility, how your immune system reacts to this, how you respond to that in different ways, and then what triggers actually set you off. But we don't really know all the relationships and exactly how we can mitigate these. So unfortunately, our gastrointestinal, our GI colleagues end up treating these with big guns like steroids and immunomodulators, and those also have their own secondary effects. So let me introduce you to one of our first topics. This is about 15, 16 years old now, the cancer stem cell hypothesis. And this is one way of looking at cancer and explaining heterogeneity. So on the left side of these slides, you can see that there is this pathway, which kind of explains how we think about sporadic colon cancers. Every cell in this pathway on the left, the A pathway, has the potential to undergo a division where each daughter cell has the capacity to form a new tumor. Contrast that to the right side of the slide, the cancer stem cell hypothesis. In this pathway, each daughter cell, they divide asymmetrically. One has the complete capacity to recapitulate the entire tumor, and the other one is a more differentiated daughter. So this is another explanation for heterogeneity in the tumors. The daughter cells, the cancer stem cell daughter cell has the capacity to recapitulate all portions of the tumor. It forms maybe less than 5% of the tumor mass, and undergoes this what's called self-renewal process. So what does that mean clinically? So the implications clinically are, and if we go down the left side pathway here, that if we have some type of drug or treatment that treats only the non-cancer stem cells, that the tumor, the bulk of the tumor would regress, but if the cancer stem cell remains, that the tumor could recur. So this is a model for late recurrences. Up here in this other column, or this other pathway, we see that if we can kill the cancer stem cells, so obviously this is the Achilles heel of all of oncology, then in theory that the tumor could not regrow and that the patient would ultimately be cured. So what we needed when we started looking was a wedge or a tool. And I remember when I was an intern, I used to be called a wedge, a simple tool. So we found our simple tool in aldehyde dehydrogenase. It's a detoxifying enzyme. It's in a family of detoxifying enzymes. They confer resistance to alkylating agents, and they cause this enzyme family converts retinol to retinoic acid. And this is a modulator of cell proliferation. And other types of cancer stem cells in the brain, so glioblastoma, in the breast, so the breast cancer, and in leukemia, this aldehyde dehydrogenase has been found to be another cancer stem cell marker. So what did we do first? We did some simple experiments. We looked at normal stem cells, or for normal colon, and we all remember from basic histology and physiology that the unit in the colon is the crypt, and they're shaped like test tubes, and the cells slough every 3 to 5 days into the lumen, and at the base of the crypt are the stem cells. So you can see here, there's a few markers here for the red or pink staining for aldehyde dehydrogenase. At the leading edge of sporadic cancers, there's also clumps of cells that can mark with this tumor marker. In colitis, this is expanded. This is, we think, because potentially of regeneration here, that these crypts are undergoing extensive regeneration. They're trying to regenerate from the bottom up to reconstitute the mucosal lining. And then this is one of my poor patients who had colitis-associated cancer, and you can see evidence of this pink-red staining throughout wherever there's pretty much epithelia marking this tumor. So we asked a question. So for sporadic colon cancer, we identified aldehyde dehydrogenase as a marker. Would this also be possible for patients with colitis or colitic dysplasia? So we took tissue from one of my patients and we implanted it subcutaneously into the flank of a mouse, just like we had done for sporadic colon cancer. And we didn't know if it would work or not, but as it was, the first case worked, and then it was a long time before anything else worked in this model. And we implanted it subcutaneously into the flank of a mouse, and miraculously it grew. We were able to split this tumor into multiple parts, expand it into multiple mice, and we were able to get tumors that grew serially from this. We were able to eventually identify these as really poorly differentiated based on histology, and then eventually we were able to learn techniques so we could carry these in vitro and culture in 3D cultures that was a wonderful tool, because then we can manipulate these ex vivo and put them back into mice and ask questions about the biology. So our next question was, do the epithelia act alone? And I told you about one member, or I mentioned one member of the microenvironment, the fibroblasts, and we'll get back to that. But other members include lymphocytes, monocytes, vascular endothelia, and they communicate through secreted factors. Some people think these are packaged in exosomes now, but certainly they're secreted into the media, including IL-2, gamma interferon, IL-1, IL-6, VEGF, and the fibroblasts have classically been associated with such chemokines and cytokines as TGF and HGF. So we asked, are there enough of these in proximity to the epithelia that they could talk to each other? So we see here immunofluorescence, and here at the base of the crypt, stained in green, are some of these aldehyde dehydrogenase stained cells, and then the stroma here is stained by smooth muscle actin, indicating these as activated fibroblasts in the stroma. And I have to say that it was probably about 5 or 10 years before I could figure out how to grow the epithelium in these non-adherent cultures as technology evolved. But every time I took a tissue and put it in culture and tried to get epithelia in the beginning, all I got was these spindle-shaped cells, which we eventually recognized to be fibroblasts. So I asked the question, are the fibroblasts from the different milieus, are they different? Do they secrete different chemokines or cytokines that we can use and might be targetable? And in fact, when we check the conditioned media of these, of the different fibroblasts isolated from cancer or colitis or normal colon, indeed there were differences. And the differences were in this chemokine called IL-8 or CXCL8. And you can see here that there's differences in the size of these little dots on this cytokine array, that colitis and cancer are much larger than that from normal colon and much larger than that from control serum. So we knew we had something here. We were absolutely able to quantify this eventually with CXCL8 directed ELISAs. CXCL8 or IL-8 has a number of roles. It has roles in migration and polarization, roles in many inflammatory pathways, roles in cell differentiation, and eventually you can see how it has all these roles in signaling, which are very happy for a tumor to have, leading to potentially proliferation, angiogenesis, migration, and metastasis. So we thought that perhaps there might be a role that these tumor stem cells might secrete some of these factors that lead to angiogenesis, for example, and then the stromal cells may also secrete chemokines or cytokines that can be targets for therapy. And indeed, there is therapy in the clinic now against the receptor for CXCL8 in breast cancer. So this is already in use in clinical trials for breast cancer. We asked some very simple questions, and this was published several years back, where could we co-inoculate some of the cancer stem cells with fibroblasts from these different environments, and would it lead to differential growth when implanted in the subcutaneous flank of an immunocompromised mouse. And indeed, in parallel to their CXCL8 expression on the cytokine array, that was how the growth was differentiated, so that when co-injected cancer stem cells plus colitic fibroblasts here, they had the greatest growth compared to cancer-associated fibroblasts, and then normal colon fibroblasts, and then here is the cancer stem cells by themselves. And could this be blocked or mitigated? Indeed it could. So we were able to do that same experiment, but only with the colon cancer initiating cells or cancer stem cells, and we placed next to it these time-release pellets that had an anti-CXCL8 or anti-IL-8 or anti-IL-6 slow timed-release tablet next to the cells, and we found that we were able to mitigate the growth response depending on whether anti-IL-6 and anti-IL-8 were present or just anti-IL-8. So this was a significant study that we reported in Cancer Research several years ago. So exogenous addition of CXCL8 to our colony assays in vitro, you can see that there's many more colonies on the right here with the and greater size with the addition of exogenous IL-8. And then we were also able to do some in vitro experiments or in vivo experiments where we were able to add exogenous IL-8 to these, and then we were able to show differential growth over the weeks that we followed the mice. In vitro, we were able to mitigate the in vitro proliferation, and this is where we either blocked we used silencing short hairpin RNA to silence IL-8 production or to silence the receptor, and we were able to mitigate the proliferation, both in vitro and then here in vivo, and we have many more of these assays coming out as we speak. So we're wondering, this strategy takes about a year to get a reagent that we can use as far as the cancer stem cells. Is there a more effective way to isolate and to propagate normal and colitic epithelia? And so here we used another tool, and from the work of some of my really famous colleagues, some at Stanford and some in the Netherlands and some in Japan, we're now able to grow primary epithelioids. So we isolate these from the primary tissues, chop them up, collagenase digest them, and then in a very specialized 3D culture, we're able to grow them. And you can see here from the normal colon, a more simple epithelium, and then from the clitics, there's a more stratified epithelium here. And we asked, what could it look like? What do these look like? And how do these compare to the primary disease? So we did some basic immunohistochemistry here. We looked at mucus secretion and ulcerative colitis versus normal. You can see a lot more mucus secretion in the normal, and we all know that, we might know that in ulcerative colitis, a lot of times during the colitic episodes, the mucus barrier is lost. There's sloughing of the mucosa with bleeding and inflammation, and so you can see here much less staining brown staining for MUC2. Lysozyme is a marker for Panna cells. It's now known in the last 10 years that the niche for growth or regrowth of epithelia is formed by Panna cells, and lysozyme is a stain for that. It's normally not present in the colon except for under regenerative circumstances, and that's what you can see here, that the colitic epithelioids have a lot more of this stain because they're trying to regenerate. So first questions, basic questions. Are there differences at the transcriptome level? So what are they making that's different, or what are they making that's similar? So we just did basic RNA-seq on these organoids to ask these questions. I mean, this has been done at the whole tissue level, and we can see in the colitic colon versus the normal colon that these kind of group separately on a principal component analysis, and that this is confirmed in the in this array here where we look at the heat maps and compare them with the normals basically lining up here and the colitics here. So definitely differences here. We looked at a volcano plot to look at the differences between expression, and the red dots here are the colitic and the gray dots here are from the normal colon. And you can see there's definitely areas with more than twofold differences in their transcriptome expression between the colitic and the normal derived epithelioids or organoids. And we could see definitely that there were several of these that really were very polarized in their expression. These are some of the colitics. This is a principal component analysis as well. And the normals. You can see these are very polarized in their expression. And so we took these 4x4 expression portfolio forward and asked about what pathways might be involved. And here are gene subsets that are upregulated. No surprise, inflammatory responses, developmental biology. Makes sense. The colitics are trying to regenerate themselves. Pathways in cancer, interestingly enough, in the colitics. And then downregulated are some other of these metabolic pathways. But having said this, that it's not surprising what is seen here. Ultimately, as we subjected this to a Ingenuity pathway analysis. Interestingly, what is the mainstay of our immunomodulatory treatment these days for IBD? It's anti-TNF. And interestingly, this pathway came up as the #1 pathway that was different, and all points, many points leading to this pathway here. And then obviously there's a bunch of metabolic pathways involved in the process, and these were also upregulated in the colitic versus the normal. So the next question we asked is, there's differences at the transcriptome level. How might these be regulated? So we asked this question looking at the epigenetics or enhancers. What are enhancers? So these are relatively new findings in the last 10 years in genetics. These enhancers are DNA motifs. They act in cis. They are binding sites for transcription factors, meaning that they help turn on downstream transcription. But they can be thousands of base pairs away, tens of thousands of base pairs away, in fact. But they do lead to certain phenotypes. In fact, they can lead to blonde hair or blonde mice by acting on a receptor at the base of the hair follicle or transcription site at the end of the hair follicle. They can lead to pediatric ependymoma. So this was actually a Nature paper from somebody that was on our floor where they found that there was no genetic findings for pediatric ependymoma. They couldn't figure out why they had it, but it was all at the epigenetic level. And even for sporadic colon cancer, we now know that there's enhancers involved here that might be able to be targeted for therapy in the future. So how do these work? How do you find them? So we can look for these by looking at where certain histone marks sit. They're marked here. And we can identify the sequences they sit to by doing chromatin immunoprecipitation, followed by deep sequencing to find out exactly what transcription factors might be upregulated or enhanced. And I'm just going to show you one example here. Remember, I showed you that staining for lysozyme before, and here we have great enhancement of transcription factors or activity for H3K27 acetylation, a marker for enhancers, by the lysosome promoter site. So that matches up with what we saw on the immunohistochemistry staining. And we're exploring some other options here. There's many transcription factors that were upregulated, but here's another one for S100P, which we published years ago might be a marker for sporadic colon cancer. And when we did further bioinformatics analysis, we see that the highest degree of sensitivity for the comparison between the colitis and the normal organoids was these two systems, not surprisingly, marking for neoplasms in the GI tract. So the gut turns over frequently, but in these previous two models, I've only really explored the epithelium. What about the mesenchyme? What about the fibroblasts? Is there a way to bring these back? And so for this, we needed another tool, and we borrowed technology from two other of my friends. Okay, so on the right here is Shinya Yamanaka. He won the Nobel Prize in 2012 for work that he did in 2006. And then was joined by a partner who had worked on reprogramming many years ago, about probably 50 years ago. So they both won the Nobel Prize, but we really borrowed the work of Shinya Yamanaka, who has a faculty appointment at another UC school, and actually started his life as a surgeon. Then he realized he could make a bigger contribution this way. Let's see. So again, returning to my friends, the fibroblasts, and what Shinya Yamanaka did in 2006 was he was able to figure out what transcription factors he had to give to a fully adult mouse fibroblast to reprogram it to pluripotency. And so we adopted his strategy, and indeed what we did was we took colon fibroblasts that we had isolated, and I showed you we could get those pretty easily by basically serendipity. And we were able to put in these transcription factors known as the Yamanaka factors. We were able to reprogram them to pluripotency. And I'll show you in the next slide how we prove that. And then using technology from another one of my friends, Jason Spence, we were able to take them through a directed differentiation pathway using a series of growth factors, first making definitive endoderm, and then making hindgut, and then making what we call induced human intestinal organoids. So this is pretty amazing, taking a fully adult cell, reprogramming it to pluripotency, and then forcing it down a path to try to make some organ. In this case, we've chosen gut. So first, when we did this, we wanted to prove that it could make all three layers of the primary germ layers. And normally we can do this making a teratoma, but our animal care and use facility would not let us grow teratomas in the mice. They thought this was inhumane. So instead, we used a series of different directed differential growth factors, and we were able to stain showing that OTX2 correlates with ectoderm, this with mesoderm, and then this with endoderm, and we were able to prove that we were able to get all three germ layers to differentiate, so that we did have pluripotency. We next decided we needed to compare what we grew to real primary tissues. So you'll see in these slides, every slide is set up similarly, where the primary tissue is from patients is on the left upper hand side, and then what we've been able to grow is on the right side. And you'll see here that you can see the primary tissues here, the colitic colon with a stratified epithelium. There's a lot of kind of infiltrated mesenchyme here. And here, control colon, simple kind of epithelium, single layer, a lot of goblet cells here, not so many goblet cells over here. And you can see here when we measured the thickness of this epithelium for the primary tissues, it was much thicker here than in the proximal or control tissues over on this, in this panel. And then we compared. These are the IHIOs, the induced human intestinal organoids. So simple columnar, and this is from 3 different patients that we started materials. And then from the colitics, we can see here we've been able to create a tool here where we recapitulate this thickness in the induced pluripotent cells, from the induced pluripotent cells. And again, when we enumerated that, it showed it here that this was twice as thick as from the normal colon. So regeneration or proliferation here, Ki67 as a marker. The colitic colon, a lot more of these brown spots indicating a lot more proliferation going on. And here, the control colon, not as much. We enumerated that here in the same way, a lot more in this distal colon compared to a proximal or control tissue. And then look at the normal induced cells. There's a lot more, not very much here. And then on the right side, here you can see a lot more proliferation, and we enumerated that and summarized that here. So great similarities so far between the colitic tissues and colitic IHIOs and the normal IHIOs and the control tissues. Mucin secretion. So I mentioned to you that in the colitic colon, as it sheds its lining, it loses its capacity to have so many goblet cells and to make mucin. We stain for that by looking for Alcian blue. Here in the normal tissues, you'll see a lot more blue, Here in the primary ulcerative colitic tissues, you see a lot less. And that phenotype is recapitulated here. Normal colon here in the top with some blue staining here at the edges, and here on the bottom with the colitics, 3 patients, 3 different patients, lack of this Alcian blue. We were able then to ask the question, would these maintain their phenotype if we put them into the flank of a mouse or in vivo? What would happen? And indeed, the phenotypes maintained. You can see these are normal, from normal IHIOs. These are from colitic ones, and you can see dominantly, you can see differences in regeneration or proliferation here with key 67 staining. You can also see an absence of mucus here from the colitic ones, and the much more mucus staining or Alcian blue staining for the ones that were generated from normal colon. CDX2 is a transcription factor marking colon, marking gut, and we'll talk about that probably in another talk in the future. So in vitro function. So one of the other questions we had is, can these react to a stimulus that's outside the little spheroids or organoids that we've generated here? So what we did here is an experiment where we took the organoids and we put with them exogenously fibroblasts isolated from the different environments. And the question is, can they respond to an external cue in this environment? And the answer is yes. They respond by having a lot more proliferation. They respond by making a lot more of our friend, CXCL8. They make a lot more, they grow a lot bigger, and they grow a lot, they make a larger number of these spheres as well. So that was a clue to us that these can respond to external cues even in the in vitro state. So again, with evidence of this phenotypic differences, would there be organized differences at the transcription level? So we asked the same question, doing an exploratory experiment, and the question was really, would these line up according to their degree of progression through the process from induced pluripotency to definitive endoderm to a bud and then a mature organoid, or would they line up depending on whether their origin was normal or colitic? And indeed, what we found was they lined up according to their stage of differentiation. So the definitive endoderms were all here on this principal component analysis, the buds were here, and the organoids were here. And indeed, the iPSCs all lined up in one cluster together. It didn't matter whether they started as colitics or normals. So in trying to analyze this, we started to try to group these by these groups definitively. So definitive endoderm, buds, organoids, induced pluripotent stem cells, according to this pathway of differentiation. But we found that there weren't really enough genes in the pathway to talk about it except for the definitive endoderm until we changed what we looked at, whether it started from normal colon or colitic colon, and then these processes or these steps from definitive endoderm to buds or buds to intestinal organoids. So it depended which pathway we started with. And then we had plenty of genes to look at to ask about different pathways that might be involved in this progression of developmental biology. We were able to categorize this using, here's an example from Revigo, and you can see here that the left is all inflammation, and you can see that there's inflammatory processes and repair processes going on. And on the right side, we can see colitis versus normal, and what's really the pathways that are really involved here are cell cycle and DNA alterations. When we look at Ingenuity pathway analysis, say what factors are really involved here, PI3 kinase pathway is one of these pathways, and in the intestinal organoid versus the bud, there are big differences between whether it's derived from colitis or from normal colon, so one difference there. Here's another pathway of analysis, and obviously there's differences here in the normal progression in SOX2 pathway, very different in the colitis. So just an example of what we're going to be analyzing going forward in this development. So what can I tell you? So I think I've convinced you that we can use stem cell-based models to interrogate the pathogenesis potentially of colitis and colitis-associated cancer eventually. The microenvironment definitely contributes to the pathogenesis of colitis and cancer. I think I've demonstrated that. And I think that new knowledge in genetics and epigenetics will lead to new interventions. And we're hoping to bring those forward to you in the next decades or two. So I'd like to end by thanking all of our collaborators between my laboratory, now at the Cleveland Clinic, friends of ours at Michigan, other friends that we've tapped at the Cleveland Clinic, including my Department of Colorectal Surgery, whose patients and operations I bother every week to get tissues, support from the NCI. We had a support also to start this project from the American Society of Colorectal Surgeons, and then a local cancer philanthropy group at our institution. So I'm open for questions.
So for the medical students in the audience, this is a career that happens after just a simple case when you scrub and ask a simple question. It launches many more questions that foster a career of investigation. So I'll start off linking inflammation. So a lot of these patients with Crohn's and colitis have varying degrees of inflammation during their life and varying risks for this. So this link in terms of ongoing chronic inflammation, like some of the other models that we see of inflammatory neoplasia, suggests that the inflammatory environment, both in terms of its longevity and acuity, are the precipient models of neoplasia, neoplastic transformation. So does that mean that the treatment for these diseases should be more at interrupting the severity or the longevity of the inflammation as a way to interact for cancer prevention?
So the answer is yes.
Oh,
You knew
okay.
that, right? Yes and yes. I think we see the same circumstances, for example, in Barrett's esophagus, and I think for many of the non-virally or non-pathogenic media that promote this pathway, we don't understand it very well. So these are opportunities for us to create tools and try to understand the pathogenesis better and then gauge our prevention. You know, it makes you wonder, though, is long-term steroids, long-term immunosuppressants, is that the right way to go? And probably it isn't. It's just that we're band-aiding the process without really understanding it.
Dr. Park.
Thank you, Emina. When I listened to your talks, I realized how little we really know, right? The more we know, the more questions we get. And to kind of echo your answer with Dr. Bull in regards to what is it? Is it the acute inflammation or the length of time? And, you know, we've all had ulcerative colitis patients who truly only had the symptoms for 2 years. And here we are at 28 now with rectal cancer. So we know it's not always the longevity. Are there other people working on potentially the microbiome part of it as a potential trigger that then creates the pathway that you've been investigating?
So the answer is yes. The NIDDK has a whole human microbiome project where they're collecting poop from people nationwide. There's some question about whether we can alter what we eat and change our microbiome. And certainly in the last 5 to 7 years, with the onset of CRISPR Cas9 technology, you know, which actually starts from a bacteria plasmid, can we use the bacteria that we have actually as a weapon to fight this off? And I think that would be the newest, latest, coolest way to deal with this and to prevent this from the beginning. Or actually, potentially someday, some decade, probably when you and I are not here anymore, using these as direct therapeutic vaccines. Yeah.
Dr. Hendon.
About 15 years ago, a group at UMass Worcester developed a sort of an interesting H. pylori mouse cancer model, and they used it to explore the issue of whether the stem cells leading to the cancers, and in fact the metaplastic gastric epithelium in these H. pylori infected mice, came from stem cells leading to the metaplastic epithelium and ultimately the cancers in these mice derived from the transplanted stem cells. And I just wondered, I could see a lot of parallels in what they did with what you started exploring in the colon. You've taken it much farther. It seems like that might be another fertile field for investigation. But I've never seen their work duplicated in another cancer model. And I'm not sure whether that's just something that's, weirdly appropriate to mice. There have been case reports in people in Japan who early in life have had bone marrow transplants with gender mismatched donors, and they get metaplasia and then gastric cancer, and the stem cells derived from the transplant, not from the person.
Right. So that phenomena is also seen, sadly, in some liver transplant patients, where they're transplanted potentially with hepatocellular carcinoma, and we have that whole question about withdrawal of immunosuppression and so forth, and there's that gender mismatch, and so sometimes we see that, which is like a huge tragedy. But you're right, and some of what their findings were interesting. One is obviously gastric cancer has that very strong relationship with H. pylori to begin with, but it also speaks to what you're saying about the bone marrow, right? Is the bone marrow is so fertile, and is there something in the organ, in the neoplasia that started, that also calls the bone marrow cells to it, with factors such as SDF1, CXCR12, for example, and I think that is also part of that. With that phenomenon, maybe why you haven't seen it reported more often or more strongly associated, is the question of the microbiome at different facilities, at different animal facilities. And so now much of the research is going to notobiotic type facilities, which aren't everywhere. I'm pretty sure you guys probably have one, with the vet school and so forth, but a notobiotic facility, so you can explore on a null background those types of questions. And I think that certainly our facility is getting a notobiotic facility. UNC has one, New York, Boston. So a lot of places are now getting these notobiotic facilities so they can ask those questions in mice. But yeah, you're right, it hasn't been picked up more. But so the question is it the bacteria themselves? Is it something with the disease process, or is it really just bone marrow trafficking? So it's hard to interrogate all those compartments.
Dr. Gingrich.
Hi, my name is Alicia. I'm one of the second-year residents. Thank you for your talk. I think the research was remarkable. From a career development standpoint, what recommendations or advice would you give to those of us considering careers as surgeon scientists, and how did you develop yours?
So I was lucky because I went from piano performance to where I am now, so it's not really the standard route. I was really poorly mentored early on in my career, and I kind of fell into this. I knew, I think probably from my time at Stanford, that I wanted to see if investigation could be part of what I want to do. But you guys have so many opportunities now. I mean, the whole health service and outcomes field has just mushroomed. So I would say figure out what questions that you're passionate about. Make sure you have alignment between what you do clinically and whatever that passion or that question is. And then you need to find great mentors and great sponsors who will give you an opportunity to do that. Not every place can have the luxury of letting somebody be released from service for time to do stuff like this, or to be able to build a team that helps you with that. I know Dr. Farmer's been really wonderful about allowing people to pursue some of these, you might call them side angles, but they're all necessary to be a truly academic institution.
Further questions? None? Hearing none, we will adjourn. adjourned. Thank you very much.
Thanks for a great visit.